This video will serve as a very brief introduction to the structure of nucleic acids, namely DNA and RNA. These molecules, of course, play very important roles within living cells, but for now we will just really focus on the structure of these types of molecules. What we see here are Watson and Crick with their model of DNA. They published their results on the structure of DNA just in 1953, so really not that long ago. But of course this had profound implications for our current understanding of biology. Let's begin with a brief look at the structure of DNA. You see a DNA molecule illustrated toward the right here. DNA really just consists of a few basic components, namely phosphate groups, sugars, in particular deoxyribose, and bases. So in our illustration, phosphate groups illustrated by the letter P, deoxyribose sugar molecules indicated by the letter S, and these various nitrogenous bases more toward the interior of the molecule. So the bases we see in DNA are adenine, guanine, cytosine, and thymine, or simply A's, G's, C's, and T's. So again, it's these bases that we see located toward the interior portion of the molecule, and it's the sequence of these bases that really are probably the more important aspect of the DNA molecule itself. If you consider the structure of DNA, we might say that a component of DNA consists of a phosphate group, a sugar, and its associated base. So you might come across the term nucleotides. So nucleotide is just that, a single phosphate group linked to a single sugar, and that sugar linked to a particular nitrogenous base, again either A's, G's, C's, or T's. So these nucleotides are linked together to form a molecule of DNA. We talk about DNA as being organized as a double helix. So you see that illustrated toward the bottom here. So notice there are two strands or two sides of each DNA molecule, and again oriented in this helical fashion like we see here. And also notice that each strand or each side has some directionality. We talk about the two strands of DNA as being anti-parallel, simply meaning that they are oriented in opposite directions. So what are we talking about here? Well, you notice these fives and these threes, these 5' and 3'. These just refer to directionality. So if you take a look on the right, this is an illustration of a phosphate, a sugar, and a base. So numbers just refer to carbons within the sugar. So if you look at this sugar molecule, there's carbon number 1. Carbon number 2 is right there. They aren't labeled, but right here is carbon number 3, hence the 3'. Here's carbon number 4, and then carbon number 5, hence the 5'. So again, we're not going to dwell on it so much at this point, but this directionality is indeed quite important, as we'll see later, because various enzymes involved in DNA replication and gene expression, they only work in a 5' to 3' fashion. But nevertheless, at this point, we just want to illustrate the anti-parallel nature of DNA molecules. DNA also displays complementary base pairing. So if you know the sequence of bases in one strand, say in a 5' to 3' direction, we have ATT, GGC, you of course can easily deduce the sequence of bases in the adjacent strand. A's always bind with T's, and G's always bind with C's. So this complementary base pairing, again, very characteristic of DNA molecules. So just a few comments about the bases, the nitrogenous bases. Of course, we say they're nitrogenous bases simply because they do indeed contain lots of nitrogen. Looking at the structure of DNA bases here, A, G, C, T, we can also talk about some of these as being purines, namely these double-ringed bases, and others as being pyrimidines, or single-ring bases. I also want to briefly point out that if you're talking about RNA, RNA does not incorporate the base thymine, instead it incorporates the base uracil, often indicated by the letter U. So again, that's just one little difference between the structure of RNA and DNA. I also would like to point out that if you look at the structure of DNA, again, we're just looking at a small portion of a DNA molecule right here, the two strands or the two sides of DNA, they're actually held together by hydrogen bonds. Hydrogen bonds that occur between the bases of adjacent strands of DNA. So if you recall, hydrogen bonds, we illustrated those previously using water molecules, but the same thing occurring here. So very important, obviously, this holds the two strands of DNA together, but fairly weak enough so that the two strands of DNA can be separated, which often has to occur anytime DNA replication takes place or anytime a gene is expressed. So let's conclude with just a few comments on the basic structure of RNA. We talked about DNA for the most part. RNA, in many ways, very similar in structure to DNA, with a few exceptions. So whereas DNA tends to be a double-stranded molecule, RNA, whether you're talking about messenger RNA or transfer RNA or ribosomal RNA, they tend to be a single-stranded molecule. Also, we pointed out that whereas DNA incorporates the base thymine, RNA uses the base uracil instead. And finally, of course, the sugars themselves, whereas DNA incorporates the sugar deoxyribose, RNA incorporates the sugar ribose. So again, structurally, these two molecules are very, very similar with few exceptions, but of course they play radically different roles in terms of information storage and information processing within cells. This concludes our brief introduction to the